I think there is a barrier around the lock when it comes to something like FDA clearance or etc. That having something that's completely open is not going to fly potentially. So what are your opinions on an openness, and then there's a point at which you lock something?
Yeah, so pertaining to the question about whether we should be using and going further into ultraplex and things like that. So I think it's been shown in the literature actually that the more you use, the more the parameters you use, the more you're gonna have granularity about a given situation. And then of course when you switch to translational medicine, when your signatures have been identified, then of course you can reduce your approach and use only those parameters which are generating the variance in the data set, which means which are therapeutically actionable. But I think it's maybe the right question would be, I mean, it's all related to cost, right? Using a lot of parameters simultaneously is going to be requiring a lot of resources, bio-IT resources as well. So if in the near future we manage to have those prices sync, maybe everyone is going to be able to employ ultraplex technologies in their research, and then reduce it as it's time to be switching gears to translational medicine.
So how do you feel about taking that kind of thing into the FDA at this point?
Yeah, so we have history with that with nanostring, because we have a breast cancer panel actually which has been FDA approved. So this is being run as it's called Prosigna. It's being run as a routine clinical diagnostics tool. And this is basically stemming from research, preliminary research that we have been carrying on together with collaborators. We started with 770 gene signature panel, and then we came up with those refined signatures that are now FDA approved. So I think it makes sense actually to be starting big and reducing the approach as students move forward towards accreditation. So.
Roberto, do you want to comment from a Fluidigm perspective in terms of that? And I've still got Ultiview here, which is great as well. So.
Well, thanks. So yeah, I mean, I pretty much agree with what has already been said. I think that now, if I have to take a look, I believe at what we see the marketplace growing really, both in the multiplexing realm, etc. I do agree with the fact that in the protein realm, we still don't exactly know what we're looking for, because it's true that protein analysis and multiplexing has not yet benefited from the scale of discovery that we've had in, for example, single-cell RNA-seq, etc., where we can just do whole transcriptome analysis, whole genome analysis, and really get everything in. But the beauty of the technologies that are coming out now is that it's actually allowing us, and like what Bernd said, to actually start to refine those sort of markers that we want to go and see. And I believe that what we're seeing now is that we are in the phase where we're starting to really try to understand exactly how many markers is enough. Because up until now, we've seen that even all of the gold standard companion diagnostic tests, think of the PD-1 assays that are out there, aren't sufficient enough to stratify patients at the moment. So now we're really in the phase where a lot of different groups, pharma companies, et cetera, are really trying to understand, okay, 1 isn't enough. Will 2 be enough? 3 be enough? 4 be enough? Apparently that's not the case, but we're going really, I think also Bernd's example is quite nice, in really trying to understand, okay, where is that limit where we can really start to go in and do that stratification? And as Alexander said, of course, cost is going to be a very big issue. At the end of the day, if we want to bring this to the clinics, it has to be something that is cost effective, can be reimbursed at the end of the day, and can really also be used to screen the patients that need to be screened.